Good morning, good afternoon, good evening, wherever you are. Welcome to Virtual Grand Rounds. Premier Education brought to you thanks to our sponsors at no cost, which is AstraZeneca, Medtronic, and McGill, the Science of Care. This week, it's a great delight for me to present Professor Darcy Wagner, who is both based in McGill as well as Lund, to continue on our transplantation theme to talk about bioengineering a human lung tissue. Darcy, over to you.
All right, so thank you for the opportunity to present our work today on trying to bioengineer new lung tissue for transplantation. So I probably don't need to introduce this much to this audience, but I just want to do a very quick recap that chronic lung diseases are a growing and very complex global health crisis. They are right now the third leading cause of death worldwide, and more or less the largest percentage of those patients are affected by COPD. This disease is caused both by genetic and environmental causes, including smoking as the number one risk factor, but also indoor and outdoor air pollution. These patients have diminished quality of life. There's really no effective therapies, mostly right now limited to symptomatic therapies. And while we traditionally think about pollution being in the form of, for example, here in Sao Paulo, due to industrial waste, we also have, of course, more recently in Montreal, we have wildfires that also have increased our amount of lung injuries that happen, and this is going to continue to increase with climate change. And so at the core of all this is that different patients are exposed to these different insults. And so in some patients that are living next to each other, the lung heals, and in other ones, maybe living next to them, they do not. And so at the core of this is that we have defective regenerative processes in these patients. And so again, just a quick recap on how the mammalian lung repairs or regenerates following injury. We know from decades of studies that there are different regional endogenous progenitor cells that line the airways in the distal lung, and that these are populations that respond after different injuries. These cells proliferate and they eventually repair and heal the lung. What we've also learned from recent single cell studies is that there are several diverse populations of these cells, including rare types of progenitor cells or aberrant progenitor cells. This is still incompletely understood, that arise during injury. And so what you can see, for example, here, that patients that have both COPD and IPF, that they lose the number of alveolar type 2 cells over time in this disease, and that is sort of directly reflected by a lack of the distal lung tissue to be able to regenerate with this loss of alveolar type 2 cells. In addition, there are new cell types that arise that we don't see in the normal lung. An example of these are so-called aberrant basaloid cells, which only appear in patients that have disease, and these seem to have an intermediate cell fate. And so again, these patients have major defects in these different stem and progenitor populations, which likely leads to the inability for these lungs to heal. And so due to this really complex landscape, presently right now for all these indications, lung transplant is really the only option or the major option at end stage for a variety of different chronic lung diseases, including also during the pandemic, as this audience knows, that there was a subpopulation of patients also transplanted that had severe COVID-19. But the major problem with lung transplantation is that there's not enough donor lungs to match demand. If you're lucky enough to get one of these rare lungs, it's complicated by the lowest transplant efficacy of all solid organs, with only about a 50% survival rate after 7 years due to the use of immunosuppression. So in order to really meet this unmet clinical need, we need to explore new options. And so there's different regenerative medicine approaches you can think about. Obviously, you could try to pharmacologically stimulate these endogenous progenitor cells. You could also try to deliver genes or cells, and in this case, delivering cells with the goal of engraftment and structural repair. There's, of course, other people focused on immunomodulation, where you try to deliver cells, for example, mesenchymal stromal cells, or even just extracellular vesicles. And there's exciting ongoing clinical trials in this space. Or the main area that my lab focuses on trying to bioengineer tissue or organs ex vivo for transplantation. So if we're able to do this, we think that obviously this would be transformative for a large number of patients. And so I just give you a few ideas of things that we're thinking on, but I'm not a surgeon. And so this audience probably has lots of other really great indications that you probably encounter in your day-to-day life that I would love to talk more about because the direction the lab is moving in is really developing custom solutions for very specific clinical problems. So obviously, the sort of catch-all would be if we could really do this on a large scale, we could increase the available donor tissue for transplantation. But I also think there's a lot of exciting potential uses in airway reconstruction, for example, in patients with stenosis, tumors that invade the airways, or example for patients with malacia. We could actually engineer, and I'm going to show you some of this, perhaps small segments of tissue that could be transplanted. I also think there's exciting opportunities to replace lung tissue in patients where there's loss of lung tissue, and just a few examples of the clinicians that I've interacted with over the last few years, right? Obviously, emphysema is one. I'm going to show you some concepts we are building on that, but also in cavitary lung disease with infection. And then, of course, if we're able to do this, there's a lot of new opportunities for modeling and studying disease because we are making human tissue. So the basic concept of ex vivo lung bioengineering is you're going to take cells. This may or may not be autologous. You're going to mix them with a scaffold that can be derived using a number of different techniques or completely manufactured. You're going to put them in a bioreactor with a hope of transplanting that tissue into patients. And for today, I'm going to focus mainly on our work on deriving scaffolds and manufacturing scaffolds that we can use for transplantation. So again, of course, this audience knows this, but the lung is extremely diverse across length scales. But what I want to remind you of is sort of the length scales that we're talking about. So obviously in the airways, we have things on the centimeter scale that are really important for function. And on the distal side, we have features that are less than, for example, 1 micron or around 1 micron, the blood gas barrier. And so this really is incredibly complex, notwithstanding also the mechanical properties that change throughout the branching architecture. And so when us and others started this journey more than 15 years ago, we wanted to start with nature's platform by using a technique called decellularization. And so in these processes, we strip away cells, but leave behind the extracellular matrix. This is an example of a mouse lung here on the bottom, and this is what an acellular scaffold looks like once you've decellularized it. This is a mouse lung. And again, just very quickly summarizing, we and others have put a variety of different cell populations back onto these scaffolds. And the main take home is that if you prepare these scaffolds correctly, a variety of different cells can actually attach and proliferate on these scaffolds. But again, what we and others have learned is that there's a very limited extent of site-specific differentiation. So what I mean by this is if I take a proximal airway cell, for example, here on the bottom, marked by keratin 5, and I put this into the distal lung tissue, it doesn't really become an alveolar type 2 cell. Now, again, I don't have time today, but there is some other work of people trying to do complex co-cultures and trying to actually push these cells a little bit more to express, for example, surfactants. But this is still really minimal, even with the best state of the art today. A few groups have undergone transplantations, so in rat and porcine models right now. The major take home is that these have been very short-term survival studies, with the exception of the study here I mentioned on the best from Joan Nichols and Joaquin Cordiella, where they did a slightly longer transplantation in porcine lungs. In both of these models of transplantation, these were single left lung pneumonectomies. The bioengineered lung had a mixture of different cell types. And the major take homes from this is that we don't really have long-term gas exchange, as you can tell. And in these bioengineered lungs, we have a lot of signs of rejection. And so there's just still a lot of work to be done in these different models. Again, a lot of my postdoc work focused on scaling this to human-sized lungs. This is just showing you that it's feasible to scale this up to human lungs. But we already started to see some things that for me started to indicate that we might want to relook at this. So the first is that we found that these scaffolds had human-specific profiles. with regard to protein. And so this means that from a manufacturing perspective, each starting point would actually be different. And this is obviously not ideal for generating reproducible products in the end that would be transplanted to patients. Of course, you could turn to xenogenic sources. So again, we and others have looked at that. What we found is that in normal porcine lungs, they actually retain some immunogenic residues, in particular galactosylated residues, which are obviously known to be a problem in other xenotransplants. But at the end of the day, many of these xenogenic sources, the lungs are really anatomically different than human lungs. And so again, with the majority of transplant surgeons that I've interacted with to date, this is maybe not really the optimal solution to move forward with. And so when I was starting my independent career, I really started to think about if we had a probable path to the clinic. And so I decided to pivot, and this is more than several years ago now, to looking at alternative techniques, again, to manufacture smaller but more specific parts of the lung. And so what we started with was 3D printing. And when I first started to give these talks in the maybe a decade ago, I had explained what 3D printing is. I don't need to do that now. But what I just want to point out to everybody is that the processes that we use for conventional 3D printing are not really compatible with cells. And so when you think about generating tissue via bioprinting, you have to think about the technique that you're going to use and also the bioink you're going to use. And what I'm going to show you on the next few slides is work that was done by two PhD students in Lund, Martina De Santis and Hani Al-Safadi. When we think about bioprinting approaches, you can really bin this into two categories. We have extrusion-based methods where we're pushing materials out, and then we have light-based methods like stereolithography, where we're using light to locally cross-link. Obviously, with these two different processes, you get extremely different resolution. So with light-based printing, you get the smallest resolution that you're able to get in bioprinting, but there are limitations that I'm going to show you. So this is an example from really some state-of-the-art work of demonstrating the potential of light-based 3D printing. What you can see here is the complexity of the types of shapes that you're able to get. And in this portion, they were demonstrating the capacity to generate vessels and even alveolar regions. Again, these are all cell-free systems I'm showing you right now, but just showing you the architecture that you're able to get with the light-based printing, and that these constructs that mimic the alveoli, that they were even able to demonstrate gas exchange. But the real challenge comes in when you're trying to incorporate cells into these processes. And so with light-based printing, there's some major limitations. The major one being is that your cells are going to be suspended in these vats of bioink. And so you have a really limited ability to spatially control where these cells are located. And so to illustrate to you how they sort of thought about using this really advanced technique you saw in the slides before, the intricacy of what they were able to generate. But when they started to include cells, it was really reduced down to a very simple model where you had fibroblasts sort of embedded in the vat of the bioink. They had an empty lumen, which we then perfused. They perfused then cells down to get sort of the airway epithelium. And so obviously there's a huge mismatch between what the technology can do with light-based printing and the complexity of the different cells that we're able to introduce. So this is why my lab went after extrusion-based printing. And again, just quickly, there are different properties when you are doing extrusion-based printing that your bioink needs to fulfill. And these are both cellular and biological. But also we have a lot of engineering demands. So again, you're going to be putting cells through needles, and so there's going to be shear forces. But afterwards, these materials need to be able to be, for example, one day surgically handled if they're going to be transplanted, so they need to be quite rigid. And again, these are general challenges. And so this is something that when we started this project, Martina and I really started to think about this list that we came up with of really minimum requirements. I also want to point out to this audience that the proangiogenic component was actually something that I was able to discuss really fortuitously with a transplant surgeon in Lund, who's Sandra Lindstedt. and really talking about the importance of this, for graft survival. And so I'm going to show you a little bit of the data because this is, of course, really important if we transplant these, that we'll be able to have them hook up to the host vasculature almost immediately. So what we're using as our material is alginate. It's derived from seaweed. And we're mixing this with a lung-specific decellularized ECM solution. And we call this our hybrid ECM solution. This is just showing you that we're able to generate hydrogels and that the ECM, when we incorporate it into the alginate, it's retained and that we get these porous structures at the end. After all this processing, we wanted to make sure that the ECM solution retained its biological activity because alginate is not known to have any biological activity towards mammalian cells. And what we could see is that obviously we had increased proliferation when we incorporate this ECM solution. So this is really encouraging, of course, that this brings enhanced biological activity. We next then turn to something called a CAM assay, a chick chorioenteric membrane assay, to assess the angiogenicity of our materials. And in this assay, you take a fertilized egg, you open up the window, you place your material on top of the egg, and then you are on the growing, developing embryo, and then you look and see how the blood vessels change over time. And as a positive control, we used a material derived from tumor lines called BME or Matrigel, and this is known to be pro-angiogenic. And what you can see is that when we incorporate our ECM solutions, not only do we have enhanced proliferation, but this incorporation of these ECM fragments also enhances the angiogenicity of our material. So this is just to show you sort of what this looks like. We built our own printers. We're now building in Montreal version 2. This is these small airways being printed by Martina and Henny. And this is just to show you that we are able to perfuse sort of the inside. These are what these airways look like. And we're also able to print things like bifurcating structures. When we put cells in these, we're able to get cell survival actually for much longer than this, greater than 1 month. This is just showing you that when we have cells with the ECM, that's similar to the data I showed you before in a more simple setup. When we print these tubes, we again have this enhanced proliferation when we incorporate the ECM into these bioinks. So we've also done a model of transplant, and this we collaborated with Paul Bourgine. And so in this model, we're using an immunocompromised animal that is deficient in FOXN1. So it's a T-cell deficient animal. And for this, we used actually a non-clinical grade of alginate, which turned out to be really interesting in hindsight. But we sort of did these pilots, and the data was so interesting that we continued to characterize it. And I think what's apparent to everybody here is that we have a foreign body response with our alginate-only material, but when we incorporate the ECM, we again see no evidence of a foreign body response. And we could even see blood vessels, really clear evidence of blood vessels growing into these grafts. This is something that was repeated multiple times. We've then tried to work to understand sort of how the incorporation of this ECM is mitigating the foreign body response. And again, this is just really highlighting one piece of data that it seems that by incorporating the ECM fragments in this, that we're able to suppress a pro-inflammatory M1 phenotype. And so to characterize the blood vessel network that grew into these transplanted constructs in more detail, we turned to an emerging microscopy technique called light sheet fluorescence microscopy. And you're able to optically clear tissues and then visualize much larger segments. These are several millimeters that we're able to look at. And what I hope you can appreciate here is that the left is the blood vessel network that we're able to visualize with for the alginate. And when we incorporate this ECM, we have this really fine vascular network that has developed. Again, this is host-derived. We did not put any vascular cells or endothelial cells into these grafts when they were transplanted. And so this just illustrates that very rapidly, this is within 28 days, that the host is able to vascularize these constructs. So finally, for this portion, we wanted to look at incorporating primary human cells. And for this, again, we collaborated with Sander Lindstedt. And so during our transplant in Lund, when the anastomosis site is resized, We're able to receive small segments, and we've optimized protocols to actually isolate the stem cells, these so-called basal cells, from the airways, from these resected airways, just a few centimeters. And so again, if anybody wants to get that technique up and going, I'm more than happy to talk to you about this. We expand these in after isolating these cells, we're able to expand them in different medias, for example, pneumocult, expansion plus medium, grow these to very large numbers to be then used in our constructs. And so we've looked at sort of, again, how these constructs or how this material can help improve differentiation. And so what we could already see when we put these human bronchial epithelial cells onto these different materials is that when we again use this ECM, that we seem to have a much accelerated differentiation, for example, mucus production in these cells. Of course, these are not completely normal looking airways. We still have some work to go, but this is just showing a cross section. So some things that we're really excited about is that it seems that the basal cells, for the most part, I'm looking, for example, at these P63 population, it stays at the basal layer, and that we have apical basal polarity. We have mucus secretion on the apical side, and we also see evidence of cilia on both actually the alginate and the hybrid constructs. So then we've gone on to 3D bioprint small human airways with this. So viability of primary cells is typically much more sensitive in bioprinting than immortalized cells. And so the ECM seems to really actually protect these primary cells during the printing. And so then for our first airway model, what we wanted to do is use these dual extrusion bioprinters so that we could actually regionally localize. So again, these are two different ink cartridges on a dual extruder bioprinter that's coordinated computationally. So we have one bioink that contains airway smooth muscle cells and another that comprises of these HBECs. We're able to print these dual layered constructs. And what you can see is we can culture them out for greater than 1 month. What we see actually after 1 month is that we have, for example, evidence of advanced differentiation of those cells. This is an example of acetylated tubulin, so a marker of ciliated cells in that inner lumen. We also don't see many cells migrating within the constructs across these different layers, but we still have a lot of work to do to further understand the functionality of these airways. So I just want to finish in the last few minutes of what cell types should we use and sort of how are we thinking about the distal lung. So in terms of what cell types we can use, I already showed this before, but again, because the progenitor cells in many of these patients are likely deranged, we have started to look at using induced pluripotent stem cells. And so this is work that I did in collaboration with AstraZeneca with a PhD student that was in my lab, Victoria Pazinski. And again, just for people that aren't familiar with iPSCs, these are where you take an adult stem cell population or adult population. This can be from fibroblasts or blood. You reprogram them with the so-called Yamanaka factors. They become these induced pluripotent stem cells, and then you direct their differentiation to the different lineages that you would like. Why these we think are ideal is that they can be made to be immune compatible, or you could have a bank of cells to do patient or donor recipient matching. We could source them from patients that don't have somatic mutations. If we go an autologous route, we know now patients that have different respiratory diseases, that the iPSCs that are derived from those have mutations. So this either would need to be gene corrected if this would be the case. And again, we can easily source and expand these cells. So this is, again, just showing summary of work, and this is based on protocols that were originally developed by Daryl Cotton's lab in Boston. And so, again, we're able to take these iPSCs through different stages of differentiation to get cells that express surfactant protein B, surfactant protein C, so really a high specificity for lung. And finally, just to show you our ongoing work. And so obviously I showed you about our work to generate airways, and now we're looking to go a bit more to larger airways, including those that have cartilage, so up to the bronchial layer. But one of the main limitations with 3D printing right now is still the resolution. So we started to ask ourselves, really, again, do we need to manufacture an entire lung for patients with COPD? So again, in most places, it's the #1 reason that the patients are transplanting, although this seems to be shifting a bit in North America, but it still remains a major clinical problem. And so here what we're trying to do is generate these microcarriers that could be about the size of the alveolus and transplanted then bronchoscopically. And so what we needed to have was sort of a manufacturing technique that can help us scale as we go across animals. So obviously the size of the alveolus in rodents is much smaller than humans. And actually, especially in rodents, this is beyond really the capability of most 3D printing technologies today. So we've been working on a number of different approaches, and I'm just going to show you one of them right now. So this is work by Sharam Parvani, who's a postdoc in the lab in Sweden. And what he's been working on is these microemulsion-based microcarriers. And so again, I'm just showing you there's a number of parameters that we have to control, but one in these is gelation speed. And what I hope you can appreciate is that we have changes in the size of these constructs that we're able to generate depending on how we control, for example, gelation speed, but also the material composition that you use also impacts this. We're able to both encapsulate cells and also coat them on the outside and culture these in bioreactors for several weeks, which is going to be needed to mature these cell types. And again, the next steps that we have ongoing for this are then to do this with a bit more control and then transplant these into animal models of emphysema. And so when we are looking forward to these transplants, how are we going to visualize the engraftment of these cells and tissue repair at a molecular level? And so there's a lot of different molecular imaging techniques that are available right now. You probably also have had some talks in these grand rounds of things like bulk or single cell or single nuclei RNA-seq, but those lack spatial information. You can also, of course, use conventional small animal imaging, but this has limited resolution. And so what we've been working on is a technique called light sheet fluorescence microscopy, as I mentioned before, but now in this case, we have labeled with antibodies. And so again, we clear the lungs and then we label, for example, here with surfactant protein C, or your cells that you've transplanted would be labeled. This is sort of a virtual bronchoscopy. And so now we're going down into the distal lung of a mouse that we've labeled. And then what you're going to see is we're going to pop out into the distal airspace, and you're going to see now these surfactant protein C positive alveolar type 2 cells. And again, this is at cellular and subcellular resolution. And so we're really able to characterize in a really complete way where these cells go and how they behave. We're also able to label other structures, for example, here the lymphatic structures, and these are things that we then can analyze in virtual reality using a software called SciGlass. So I just want to finish up and hopefully have a few moments for questions. I hope I've convinced you that 3D printing could emerge as a potential option for generating transplantable tissue, but there's still a long way to go. We have a lot of work to do with bioink development and also to scale these up to clinically relevant sizes. We and others are working on expanding the number of cells, but there's still open questions of what cell types we should use and how do we also culture multicellular constructs ex vivo. And just important to really highlight to everybody that no major functional regeneration has occurred in vivo using any bioengineered tissue. So thank you to the lab both in Sweden and the growing lab in Montreal. Thank you for your attention, and I'm happy to take any questions.
Amazing. Thank you so much, Darcy. That's like mind-blowing work. So, and also thank you so much for being up at 3 a.m. to speak to us today.
No problem.
Perhaps I'm going to start by asking, from a time frame point of view, let's say if your research is successful, what kind of time frame do you reckon it would be possible for us to actually implant bioengineered lungs?
I mean, so the second project that I showed you right now is actually it's a newer project. I mean, we've been working on the concept for 5 or 6 years, but it's really, I think, gaining momentum in the lab. And I think from a surgical perspective, if we really can administer these small units bronchoscopically, this obviously opens up lots of possibilities. So hurdles we're kind of looking at there is, identifying again, if we're going to use primary iPSCs for these types of therapies, we have to convert these protocols to GMP protocols. We've already started work on identifying potential bioreactors to do that with and companies that might be able to help us with that. For the biomaterials, there is some clinical sources, GMP grade sources of alginate that we could use. The materials that we are manufacturing, though, we have to go through that regulatory process ourselves. So we have not started that conversation with Health Canada yet, but I hope to do that quite soon because we've made quite some good progress with that. So I hate to give timeframes, right? But I mean, this could be, within a 5 to 10 year time frame, I think, for those technologies. But we'll see. You never know how fast regulatory things work. But I think if the animal models go well, and we're going to do that with clinical grade material, then I think hopefully soon. The bioprinting I think is a little bit farther away. We have some work on mechanical properties that need to really be improved there.
Thank you. Question from Professor Paul Bass, who's asking, when you seed the cells in the culture and the cells are growing to actually fit the scaffold that you have, how do you control when the cells stop dividing? Presumably, normal cells, would they stop after a certain point?
Yeah, so I mean, a stem or progenitor cell by definition should have the capacity to proliferate, right? So even in vivo, these cells should have that capacity. So the way that we control this is by the growth factors that we include in the media. So in the early stages, we have really a pro-growth media to really expand the cells on these constructs. So they're seeded not at high density. If you have both for the bioprinting or for the small alveolar tissue, if you have too high density, it's very difficult actually to manufacture. So you'll actually get like needle clogging, for example. So we put them in a lower cell state, lower cell concentration, proliferate them, and then you shift them to a pro-differentiation medium where actually proliferation should stop. But remember that when we transplant these, they should actually retain their proliferative capacity because if there's an injury or those patients get a viral infection or again pollution and they're exposed to some injury, those cells should be able to respond and proliferate under the normal cues that normally immune cells or neighboring injuring cells provide to those stem or progenitor cells.
Thank you. Can I ask, the blood vessels, at some point, you'll need to revascularize the scaffold as well. Do you envisage that the actual cells will start forming capillaries or recanalizing to be able to allow blood flow through them?
So yes, so again, the what I showed here, we didn't actually, the graphs were small enough that we were able to get full angiogenesis just through the ingrowth from the host. Since moving to Canada, I've been collaborating with somebody named Sarah Vasconcelos. If you need a name for another person in the future, maybe that could be interesting for you guys to invite for these virtual grand rounds. What she's working on is transplanting microvessels and then understanding actually how the transplanted microvessels anastomose with the host. And so we've been doing some work with her with light sheet, and it's really interesting actually to see sort of how these vessels anastomose, right, between the host and the recipient. And so I think you could think about printing some rudimentary blood vessel network, which then could anastomose with the self-anastomose, basically with the host and some of the really pioneering work that she's shown, that if these vessels are competent, that they're secreting factors that recruit in from the recipient.
That's incredible. Congratulations so much on pioneering this work and, making the future, potential possible for many, many patients around this planet. Next week, we're going to talk about AI in thoracic surgery. And once more, thank you, Darcy, for waking up 3 a.m. to speak to us. It's a fantastic talk today.
No problem. It was great to meet all of you, and don't hesitate to reach out if I can be of help.
Thank you. Bye-bye.
Bye.